Hi guys, it's Mrs. Shannon, and I'm going to do the notes that we would normally take in class. For this section, I'm going to finish them up with two different screencasts. We've done the first part of these slides last week. Actually, we did them in class up through mitosis. We talked about the cell division. We talked about different regulatory factors. We had internal signaling. We had external signaling that sort of triggers cells to be able to go into cell division and to put the brakes on cell division if need be. We talked about the different checkpoints. We did a POGO. We looked at HHMI. Some of you did at that eukaryotic cell and the cell cycle activity to investigate how cells move from one stage to another stage. And we briefly went through the stages of PMAT in mitosis. So what we're going to do here is this first screencast is going to talk about meiosis and some chromosomal abnormalities. The second screencast will focus a little bit on results of this variation through meiosis by looking at genetics, specifically Mendelian genetics, and a little bit of the non-Mendelian inheritance as well. Now, if it goes a little fast, please refer back to your book or the test prep book. It is assumed at this point that you have a foundational knowledge of genetics and meiosis. Even if it's not fresh, hopefully this will trigger a lot of memories. If not, again, please make sure that you go through everything that you have when it comes to resources available to you. So we're going to start here at meiosis. I believe it was slide number 37 in our cell division and genetics slide presentation. There's also the David Nufke Prezi, so feel free to go through that, see if it goes into more detail than what I'm giving you here. So when we're looking at meiosis, it's considered a reduction division, meaning that we have fewer chromosomes than what we started with. In fact, we have fewer chromosomes and we have we have fewer chromosomes per cell, but we also have cells that are genetically unique and different from one another. And so here, if we had a parent cell here, you'll notice that there are two homologous pairs of chromosomes. And what we mean by that is here we'll call blue dad and red mom. And so in a cell, there's usually two copies of each chromosome. The longer one here we'll call A, and we have one copy for dad and one copy for mom. Same thing with the second chromosome here. During normal cell activity, this is what it looks like. Now remember, all cells have 23 in number of chromosomes. The only cells that are going to go through meiosis are our gamete-producing cells. So those in our reproductive organs for our males and females. And so we have a cell just like any other cell. And what's going to happen is during replication, we're going to make homologous chromosomes. And homologous chromosomes, here you can see the mother chromosome that has replicated during S phase and the father chromosome that has replicated during S phase. Now, this little difference in color I'll go over on the next slide. But we still make replicated chromosomes, or these are actually called chromatids. Replicated chromatids. And they are going to divide. During the first division, we end up with two intact chromosomes here. Whoopsie. And then during the second division, these chromosomes are going to pull apart. The chromatids are each going to go to opposite sides, and we have the isolated chromatids here. So the resulting cells are not identical. They are different from the parent cell. We have 4 instead of 2, as in mitosis, and you'll see that there's a lot of genetic variation going on, and there's a couple of reasons for that. Meiosis is required for sexual reproduction, and intuitively that should make sense to you, because if we don't reduce the number of chromosomes, which is what meiosis does in a gamete, when the gametes fuse during fertilization, you are going to end up doubling the number. And if we haven't reduced it before we double it, we're going to end up with fertilized cells that have twice as many chromosomes as the parents, and from there, twice as many, and so forth. So keep that in mind as to why it's needed in the first place. Now, this is a fuzzy picture, so I'm going to ask you guys to refer back to the pictures in your text or in the test prep book. But here you can start with a normal cell, and you can go through the first phase of meiosis. Same thing, PMAT, prophase, metaphase, anaphase, telophase. But then instead of going through the cell cycle and replicating again, it jumps straight into prophase 2, metaphase 2, anaphase 2, telophase 2. Now, those different colors on the chromosomes where it looked like red and blue had swapped parts of it, that's due to something that's unique within meiosis, and that's called crossing over. It happens in prophase 1, and what happens is these homologous chromosomes become so close to one another. So we have our one from our mom. Remember, it's already been replicated, so we have the first chromatid, the replicated second chromatid, and the same thing from this one over here. And what happens is they get really close together, close enough that they actually begin to touch. And there is a synapse that occurs called a chiasma. And this transfer of information at the chiasmata allows DNA to sort of swap back and forth. And so some of this from the mom chromosome will swap over to the dad and vice versa. It does not happen in mitosis. It only happens in meiosis. But then later on, when they start to pull apart, it's going to take some of this unique DNA that's now found on this chromatid with it. So I'm going to just watch this video. I'm going to play it for you guys. I want you guys to watch it. I said most of the images, that image that we had was kind of cloudy, but if you go into your test prep book, you can see meiosis explained and set out really well. This is a 3-minute video that covers it. You won't have to identify which stage of meiosis you're in, but you need to understand how genetic variation can occur and when it will occur in meiosis.
One more time.
Dana, hurry. We have to call up soon.
Okay, so here's one that's been replicated. We'll call it the dad.
Replicating as fast as I can.
And here we'll say it's from the mom.
Quick, call up.
Okay.
Call it, dad. Where to do my business right? And the big one is almost done. Once it started, partners do their bow. Do see the crossover now. Help me live with your left hand. Around the lane.
Okay, so you can see this chiasma forming where they actually touch, and that's where the genetic material is going to be swapping during crossing over. Okay, so the nuclear envelope's gone. They've crossed over during prophase. They're attached to spindle fibers. And see, we have a meeting in the middle. Now anaphase, they separate them. Notice they're still in they're still in chromosomes. The sister chromatids are still attached. Okay, so now we have 2 more separate sets of division. Each one's going to undergo another set of PMAT. Okay, and this is where the actual sister chromatids are going to start being pulled apart in this anaphase. So this is this part that's more similar to what mitosis was.
This is just your normal thing with some lucky appropriate. Rest a while, otherwise do what you remember around you.
Okay, and so you can see these gametes now forming. We just need to pinch off and do cytokinesis.
out the ones that treat cytoplasm will divide where gametes form with you inside. And if we zoom out, you will find many more the same kind. If you're wondering what's the use, also you get reproduce.
So yes, it's cheesy. One thing I did want to point out to you is they did talk about the coiling from interphase. Remember, before prophase began, it actually coiled down. There was a line about it coiling down. So while it does condense during synthesis, during the replication, it condenses and it spirals down, coils down even more before cell division occurs. And that helps keep it separated, keeps it together during cell division process. But notice, and hopefully you saw that after division had occurred, after meiosis 2 was complete, then the chromatids themselves started to decondense, if that's a word. They started to unravel and become looser, and that's the normal state that it will be in. It only condenses for cell division. And so we'll go on then to the next slide. Also, it wasn't shown there, but hopefully notice that there is something called independent assortment going on. This is another attribute of meiosis that sort of leads to genetic variation. So we have crossing over during prophase 1, and we have independent assortment happening as well during metaphase 1. So during metaphase 1, what happens is they line up at the plate, the homologous pairs. They're still attached to their sister chromatids. But the way they line up is sort of random. The alignment of one pair has no effect on any other pair. They're all independent of one another. And what that means is that as they start to separate and go through meiosis 2 and then form the gametes, that you end up with a myriad of different combinations of chromosomes that are together. The events of meiosis in the sexual life cycle generate a tremendous amount of variation. Every organism that is produced is genetically unique. The prezi itself goes through the combinations. To figure out how many combinations that you actually have of gametes, it's 2 to the nth power, meaning you take it 2 to however, whatever the haploid number of chromosomes actually is to determine how many possible gametes are produced. And so if you do that for 2 to the 23rd, I believe it's like 7 trillion, or is it 8 trillion different combinations. But then you think that's just for one gamete. Now you've got to factor in that when you have 2 gametes coming together, each with 2 to the 23rd possibility combinations, then you get almost an infinite possibility of combinations of offspring when they do actually fertilize. That's not even taking into account the crossing over business where some of the DNA was swapped along the way. Oh, and here we go. So variation due to independent assortment is 2 to the n. N equals the number of homologous pairs. And so we have 8 million possible combinations. I said trillion, I think I was thinking down here. 8 million possible combinations. You multiply that by the 2 gametes and you have 70, I think it's trillion. Is that many zeros? Combinations in humans. And then you add in the variations due to crossing over, and it's an infinite amount. So large, tremendous amount of variation. Remember prophase 1, crossing over. We have independent assortment during metaphase 1. We have fertilization. Those 3 areas are what leads to the variation in individuals in a population. So that's sort of the overview of meiosis and how these variations, how these different chromosomes end up in different gametes and then onto different organisms. But now we're going to look at see what happens when there are errors in the process. When there's errors in this chromosomes being passed on to new offspring during fertilization. And so one chromosomal error that's common is called non-disjunction. So mistakes during meiosis happen. And if the chromosomes fail to separate, then you'll end up with gametes that have either more or less than the number of chromosomes that it should. And it depends on where the non-disjunction occurs, if it happens during the first round of meiosis or it happens during the second round of meiosis. And so here you can see that the first round happens okay. During the second round, during meiosis, we have our eggs forming. I should say that. The eggs forming right here. We have our new gametes. We have our new egg cells, daughters right here. These form correctly. However, during non-disjunction in this second one, the second round here, we end up with an egg with 0 chromosomes and an egg with twice as many. And so when fertilized with sperm, you can see that the number of chromosomes are going to vary. In this case, non-disjunction occurred in the first round of meiosis, and so you end up with egg cells with no chromosomes of a particular chromosome number in that egg cell. Often these are fatal to a developing organism. Miscarriages arise, it doesn't proceed on. There's also specific ones that do. Trisomy 21 is non-disjunction of the 21st chromosome. And so in this case, you can see an individual's karyotype, and a karyotype is just a picture of their chromosomes. And here we have an extra copy of the 21st chromosome. This is an example of non-disjunction. We have an extra 21st chromosome. There were 2 in one of the gametes. When they fertilized, then ended up with 3. Trisomy 21 may not be lethal. It does have disorders that are associated with it, because anything along this chromosome, any genes along this chromosome are going to be influenced by this extra chromosome that's here. Fortunately for 21, it is often the quality of life. Disorders affect the quality of life, the effects of this extra chromosome. But it does present in like severe retardation, facial deformities, developmental delays, etc. Not all of them are so lucky. Edwards syndrome is trisomy 18. For some of you, you know that a couple of years ago we were pregnant with our 4th daughter. She was diagnosed with trisomy 18. She did not make it to term. For those diagnosed with trisomy 18, it is considered lethal and it's not compatible with life. Only 1% of offspring actually make it to birth. And out of that 1%, only 10% of those offspring will make it. 10% of the 1% will actually make it to their first birthday. And that's just because of the genes that are affected with that particular chromosome. Now, sometimes you have non-disjunction with your sex chromosomes. Okay, so the chromosomes that we were looking at, 18 and 21, I should point this out. They're autosomes. Autosomes are any of your chromosomes that are not the sex chromosomes. They are called autosomes, A-U-T-O-S-O-M-E-S. Our sex chromosomes are X and Y. And so we can have non-disjunction with this. And in this case, we had an XX individual, an XXY individual. And this came about because of non-disjunction with either the XY or with the non-disjunction of the X chromosomes. Now, in this case, what's going to happen is that the person will be sterile. They will be a male, but they will have like budding breasts, for instance, and different physical symptoms that might mistake them for a female. Now, for any non-disjunction or any other chromosomal abnormalities, there are options ahead of time to know about this, and this leads to genetic testing, which a lot of pregnancies undergo. However, even though it's available, there are ethical questions that go along with it, which we'll talk about another time. Now, besides non-disjunction, there's a couple of other ways that abnormalities arise, and they have to do with the chromosomes themselves being mutated during replication and not being caught. So replication errors, and we'll talk about replication second semester, but we're talking about deletions in the DNA inversions where it's switched around, swapped around, translocations where we'll have genes from like one chromosome, like chromosome 2, being located and added onto chromosome 3. That's a translocation. And so there's different options here, or there's different ways that during DNA replication, errors can occur as well that will be carried with the chromosome and lead to genetic disorders. So we're going to stop here with this first round of screencast notes. Make sure you're familiar with meiosis, how it's same and different than mitosis. Also, understand where genetic variation can occur, how the genetic variation occurs, and what the results are. Note different areas that chromosome abnormalities can occur, and then lead to disorders, so like the non-disjunction or issues in DNA replication, and be able to describe some of the effects of that. It's also a good idea, and I've said this before, to have examples in your toolkit. So have examples. When I say toolkit, this is your academic toolkit. Have examples of non-disjunction. There's some examples in the Prezi of what happens for translocations. And so you'll want to go through the Prezi perhaps to look at that. You'll want to see examples of crossing over. So get into the habit of getting these specific, they call them illustrative examples, down for each event that we kind of come across. So the next screencast is going to go through the genetics, the Mendelian genetics, and this is kind of what's displayed. Okay, so we've had meiosis occur, we've had a cell, we've had a sperm and an egg come together, fertilization has occurred, mitosis has occurred, the organism begins to grow, and now we're going to look and see at what kind of traits these genes are going to end up displaying.